Welcome to Chapter 7 of Microbiology. This chapter really is getting into the heart of microbial terms, discussing what are the distinct nutrients that are necessary for different types of life forms. Additionally, it discusses the ecology or the ecological systems and associations between microbes, their host, and microbes between other distinct microbes within the same environment. Likewise, we'll conclude this talk discussing bacterial growth, and distinctly you'll get to know a bit more about the growth curve that's shown here on the front slide. So microbial ecosystems are indeed biofilms. So biofilms are typically an organization of different types of bacteria, different types of other microbes that have created these associations with one another. And essentially, my analysis in this, when I discuss this in actual live classes, we talk about, first of all, if you're going to go out to the bar or you're going to go out, to some unknown event, females typically do not go alone. And I want you to reflect for a moment, why do you not go alone? First of all, you probably thought, well, there's safety in numbers. Second of all, you may have reflected upon yourself and thought, well, it's kind of creepy. Or maybe you do go alone and you just, feel very confident and have no issues. But there are safety in numbers, and so even microbes exhibit this type of property. And they do so by forming biofilms, orchestrated virulence factors where they work together for the same good and goal of an entire community. And so how do they orchestrate this community? They do so by sending out quorum-sensing proteins. So you initially have the colonization of a free-living microbe to a cellular surface. The microbe, once it has began doubling and created a sufficient quantity of daughter cells or has association with other microbes within that environment, will start sending out distinct inducer proteins or inducer molecules. These inducer molecules are probing for density. Once then the probing for density, they send out quorum sensing proteins, and these quorum sensing proteins allow these microbes to orchestrate their valence factors, work together, and create a new ecosystem. And so why exactly is this so difficult? Why do we care about biofilms in a clinical context? Well, we care about biofilms in a clinical context because biofilms are very, very difficult to treat. So these polymicrobial infections, when you go to treat with an antibiotic that is distinctly specific for peptidoglycan, which is only present on gram-negative cell walls, you're posing a risk because you may have an entire layer of gram-negative bacteria covering the gram-positive bacteria before you ever get to that point. Likewise, you may have, ecological associations with yeast or other microbes. And those associations, when they're layering on top of one another, it's much more difficult for our standard therapies and treatments to actually get rid of those microbes from the surface. And so biofilms are a huge concern for hospitals. We think about going back to the catheter. So if you've got a catheter and you've got a polymicrobial infection on that catheter, your patient actually is at a greater risk for succumbing to the infection because of these orchestrated efforts to exhibit virulence factors altogether. And so microbes and host, we see that the microbes are highly adaptable. We have become a society that is highly reliant on antibiotics. And this reliance on antibiotics is actually having a very negative effect. So most illnesses that we have in today's society, we could typically resolve with a normal immune response. We give antibiotics strictly for the purpose of preventing long-term sequelae or long-term effects of disease. But when we give antibiotics, they also have this unfortunate benefit of impacting our normal microbial flora. And so when we alter our normal microbial flora, we're now creating not only additional space for these antibiotic-resistant microbes to outgrow, we're creating a dysregulation along our normal body. And so if you think about whether or not we should go and ask for antibiotics for bacterial infections that, our immune system could readily cover, My advice is actually no. I think we definitely need to change our tactic, and we need to work on reducing antibiotic resistance, because antibiotic resistance is emerging because when we have microbes that can replicate very, very quickly, if you happen to have one of those microbes that picks up a resistance gene, and particularly is resistant to methicillin, such in the case of MRSA, or resistant to vancomycin in the case of vancomycin-resistant enterococcus. When you have these sorts of microbes, they're more likely to outcompete the standard bacteria that is in your body, and so your normal flora is then replaced with an antibiotic-resistant strain that is, potentially would have been just regular normal flora. So the competition going down increases the infections that we experience from these antibiotic-resistant strains. And so if you would reflect for a moment, how are the ways that we are creating this antibiotic resistance nightmare? One of the ways that I can think of is, well, do you finish your medication fully? Or do you stop taking your antibiotic once you feel better? And most of you may have answered, well, no, actually, as soon as I start feeling better, I tend to forget to take my antibiotics. So that is patient compliance. So patient compliance with antibiotics is not very good. Typically, patients, once they feel better, they stop taking the antibiotic, and that poses the risk that if there are any of the microbes that have not died, if they've picked up a resistance gene, now you have a space where there's little competition and the bacteria can outgrow. Likewise, if we think about how we dispose our antibiotics, and so reflect on disposal. You probably have heard about someone at some point in time flushing antibiotics down the drain. This is really, really improper. You should take any excess medicines back to the pharmacy and let the pharmacy dispose of those through proper channels. And so when we are putting antibiotics into our water supply because of poor waste removal, this is increasing the access of microbes that just happen to be in the water, such as Escherichia coli, to pick up exposure to the antibiotics and have a longer chance to mutate and then pick up a resistance gene that allows it to be selected against. Furthermore, another thing to consider, a lot of the foods that we eat on a day-to-day basis have antibiotics. They've been taken from animals that have been given antibiotics, or we have milk where the cows have been provided antibiotics, or chickens that are dosed with antibiotics to prevent low-level infections. And so our processing mechanisms for food production plants are also contributing to this issue, and you'll learn a bit more about this in one of your homework assignments for this semester. And so the other thing I want to draw your attention to is the fact that antibiotics are drying up. We are having fewer antibiotics produced each year. And so from 2008 to 2011, fewer than 3 antibiotics were sent back out on the market. And so eventually we're going to reach a point where we have no other antibiotic therapies that are readily accessible for our society. So ecological associations, when we look at this chart, I want you to break it down thinking about symbiotic, meaning these are organisms that live in the same environment or within close associations to one another. So for example, mutualism is when you have a microbe that is completely dependent on the host. And in this case, the microbe benefits as well as the host. So an excellent example of this is a riboflavin bacteria producing glycoside hydrolase to digest milk proteins. And so these riboflavin bacteria that produce glycoside hydrolase exhibit a mutualism with the host, allowing the digestion of milk when we couldn't have done that in the first place. Likewise, if we think at commensalism, so commensals are bacteria that live on the host. The commensal does benefit by having that harbored environment, but the host is not harmed. So the host does not benefit, but the host is not harmed. If you think about parasitism parasitism is actually that the microbe must live on the host. We've discussed this term before in chapter 1. And so parasitism, the microbe is on the host and only the parasite benefits. The host is always harmed. And so these are all symbiotic because they're living in close association, versus non-symbiotic are by free living organisms. So by free living, these are organisms that do not require one another. There's not a host-parasite relation. There's not a host-commensal relationship. It is indeed two individual microbes that happen to be in the same environment. So two different bacteria that do not need one another to survive. And so synergism is when these members cooperate and they share nutrients. So for example, if you have a microbe that can do nitrogen fixation and you have a microbe that can digest another, necessary component, and because of those digestions, everyone has usable nutrients, that is a synergistic relationship. An excellent analogy for microbiology class, if you've joined a study group, if everyone is working together to review the notes, make note cards, share good information, you are doing a synergistic relationship with your classmates to make everyone more successful in this class. Alternatively, antagonism is when you have members that are inhibiting or destroying the livelihood of another microorganism. And so antagonism would be if you were particularly sabotaging a classmate during an exam. Antagonism is most classically defined when you have a microbe that produces a chemical inhibitor that will block the growth of another microbe that would compete for space in the same environment. And that is exactly how we have isolated a lot of our antibiotics. So this is the breakdown of these ecological associations. So it's just giving you the same information and a few additional slides discussing it a bit further. And so synergistic, again, working together for the good of the whole. Versus antagonism, it is how we got our antibiotics. So you can see here there is some chemical component that is inhibitory to the growth of these microbes on this distinctive petri dish. So the nutritional requirements of microbes rely on either a carbon source or an energy source. So we're first going to begin discussing the carbon sources. So microbes that are considered heterotrophs, or organisms that are considered heterotrophs, must digest organic molecules such as proteins, carbohydrates, lipids, and nucleic acids to obtain carbon, right? So to distinctly obtain carbon. In contrast, however, an autotroph can use carbon dioxide and can break carbon dioxide down to obtain its carbon source. So plants are autotrophs because they do not require the digestion of other organisms to obtain carbon. Now, the other nutritional requirement is that of energy. And so the energy currency of a lot of cells is glucose. So glucose can be extracted from ultimately, the best source is carbohydrates, but we can irrevocably obtain glucose from other metabolic processes. And so glucose, when it is digested down through the process of glycolysis, the output of that chemical reaction is ATP, adenosine triphosphate. And so the first step shown here, the glycolysis step, will provide you with a certain amount of ATP for usage in the next subsequent reactions. And so shown here is glycolysis breaking things down to pyruvate, to the TCA cycle, and then going into oxidative phosphorylation. And so in other terms, the TCA cycle is the Krebs cycle, and oxidative phosphorylation is the electron transport chain. And so glycolysis takes place in the presence or absence of oxygen. So it can take place in either case. And so glycolysis breaks down glucose, and it takes that product of pyruvate and it puts it into the TCA cycle. And at the result of the glycolytic pathway, you end up having the glycolysis take place, shown here in the cytoplasm. And glycolysis will give you a net yield of 2 ATP. Upon moving to the Krebs cycle, the TCA cycle, you have now a requirement for oxygen. This oxygen requirement means that if oxygen is not present, you could not go through the TCA cycle and finish this catalytic cascade. And so the TCA cycle requires oxygen. It's going to take place in the mitochondria, and it's going to give you a net of 2 ATP. At the conclusion of this cycle, then you're going to move into the electron transport chain or oxidative phosphorylation. And this process is really the energy store for the mammalian cell. And so oxidative phosphorylation then is going to provide us with 30 to 32 ATP, depending on efficiency. So a lot of the microbes that we'll be dealing with are chemotrophs. You yourself are a chemotroph. Alternatively, plants are phototrophs. They're able to take carbon dioxide through the Calvin cycle and convert sun into usable energy due to the process of photosynthesis. So every organism tends to have a need for distinct growth factors. These are factors that must be obtained through a various nutrient. An excellent example of this is vitamin C. Vitamin C is required to prevent the manifestations of things like scurvy. And so typically back in not prehistoric times, but back in the times of Christopher Columbus, you would hear about individuals who were on those boats for long periods of time that would develop scurvy. And so they found that they could pack limes and lemons and citrus fruits to help prevent this, because the people on the boat were lacking a large quantity of vitamin C. So heterotrophs tend to break down into two distinctive categories. Saprobes being they are free-living microorganisms that can feed from other dead organisms. And so these can be opportunistic pathogens, so microbes that are not always pathogenic but become pathogenic under certain circumstances. Or it could be that of a facultative parasite. So facultative parasites mean that the parasitic microbe is not always parasitic. So anytime we have the term facultative before a word, that means that this microbe is not always parasitic but can become parasitic. So distinct parasites are almost always pathogens, and they are obligate. They derive their nutrients always from the host. And so living on the host, they must be in that situation. So obligate means they must live on the host. They cannot live outside of the host. Versus a facultative parasite could live in a free-living environment such as water, but then also thrive in a host. An excellent example of a facultative parasite would be a worm infection. So I would like to draw your attention to the fact that you were talking about the obtaining of carbon as well as energy. So if an organism degrades organic molecules to get carbon and energy, right? When we think of that, then these guys are a chemoheterotroph. The transport and movement of chemicals across the cell membrane take place by various passive transport mechanisms and various active transport mechanisms. When we talk about passive transport mechanisms, diffusion is the movement of a solute. Right? So a solute is either glucose, salt, whatever is dissolved in water, compared to passive transport for osmosis is the diffusion of water. So it's water moving from a high concentration to a low concentration. Diffusion is the movement of solute from a high concentration to a low concentration. So it is very important to remember that solute is what is dissolved in water. The combination of solute and solvent is what makes a solution. So shown here, you can see that this is indeed a solution. It appears that a sugar cube has been dissolved in water. And so now you have a solution of water and solute, right? So potentially glucose. And then facilitative diffusion essentially is talking about a protein carrier. So the use of a protein carrier to get molecules from the high concentration to the low concentration. So passive transport does not require energy, so there is no need for ATP. However, active transport does require energy. So ATP must be mentioned in any type of active transport pathways. The energy used is going to move the specific molecule in a gradient-independent fashion. So often, gradient-independent means it's going to move from a low concentration of solute to a high concentration of solute. We do the process of endocytosis, bringing items into a cell, exocytosis, the pushing of items out of a cell, or penocytosis, meaning the drinking of fluids into a cell, as well as phagocytosis, the consumption of other organisms into a cell. And so these are all active transport processes. They are irrespective of the gradient to a concentration that is high to a low concentration. And so when we think of osmosis, likewise, osmosis is the movement of water molecules from a high concentration to low concentration. So it's a passive transport. And so when we think about the movement of water, we often think about the distinct tonicity of the solution surrounding that molecule. And so this is a cell. When a cell is placed into an isotonic solution, so iso meaning same, when it's placed into that isotonic solution, that means that the concentration of water on the outside of the cell is exactly the same as the concentration of water on the inside of the cell. As a consequence of these being equal, the movement of water is the same going in as it is going out. Conversely, if you have a hypotonic solution, so an example of a hypotonic solution would be 100% water. However, on the inside of the cell, it's most likely a lot less than 100% water. It's probably something like, maybe 80% water, so 20% other solutes. And so because there's more water on the outside of the cell, the higher concentration of water, being 100%, will move toward the concentration of water, which is only 80%, until it's equal. Unfortunately, what that can do for us, it can cause our cells to burst. Plant cells, however, have adapted unique cell walls and unique gradients that allow the pickup of 100% water without damaging the cell and maintaining turgor pressure. Our animal cells do not have this adapted mechanism, which is why if you were to accidentally give a patient an IV of 100% water, you could actually cause their cells to burst, and you could potentially cause a complete loss of a patient due to all of the red blood cells lysing. In the case of a hypertonic solution, if you've ever swallowed a bit of salt water from the ocean or gargled salt water, you know that it immediately causes all the fluid in your cells and your mouth to dry completely out. And so hypertonic solutions, hypertonic meaning high tonicity, will have a very high concentration of salt or glucose or other things dissolved in the solution. And so in this scenario, when you're thinking of hypertonic, the quantity of water is actually quite low. And so a hypertonic solution maybe has 60% glucose and only 40% water. And so when you have a hypertonic solution on the outside, but say you still have that 80% solution of solute and solvent on the inside of the cell, it causes the water to rush back out, because on the outside there's literally only 60% water. On the inside there's still 80% water, and the 80 will move to the 60% until it finds homeostasis. So that will cause your cells to crenate, or the cell will undergo a process called plasmolysis. Facilitated diffusion is passive transport when you have a protein carrier involved. So facilitated diffusion is the movement of molecules from outside of the cell to inside of the cell. There are potassium transporters that do this process. So potassium transport can be passively diffused from the outside of the cell through a protein channel into the inside of the cell. So carrier-mediated transports can take place without energy, or they could be also active transports. And so active transports require ATP to facilitate the movement of a solute across the protein gradient. And so, for example, active transport takes place with sodium. Sodium is actively pumped out of the host cell into the extracellular space or into the intracellular space, depending on the type of ion. So group translocation, in some cases there is group transport that is facilitated. An example of group transport is potassium and sodium co-transport. And so as one ion goes in, the other ion can go out. Or group translocation, another example of that would be that of glucose. So glucose and insulin are co-transported across the membrane, and so insulin is required for the uptake of glucose. So here's an example problem. And so for cell A, if cell A has 60% glucose and it is placed in a solution with 15% glucose, and the cell membrane is permeable to the solvent. So my first question for you, what is the solvent? Hopefully you answered that the solvent is water. So the quick answer for that then, if you know that the solvent is water, that is it is indeed osmosis that is taking place. So now we take the same problem and we're going to ask ourselves, which way will water move? And so to set this up, you need to know the information. So on the outside of the cell, there's 60% glucose, which leaves 40% water. What is the solvent? The solvent is water. And so the 15% glucose on the inside of the cell has 85% water. So take a moment and figure out where the highest quantity of water is located. So you can see that the 15% glucose is located in the solution outside of the cell. So the 15% glucose means that there's more water on the outside of the cell than there is in. So which way will water move if you're doing passive transport, which is osmosis? Into the cell. So we continue with example problem 1, and we talk about the movement of water. So we know that water is moving in. So our question is, the solution on the outside of the cell, is it hypertonic, hypotonic, or isotonic? It is hypotonic. And it's hypotonic because there is less tonicity, meaning there's less solute on the outside of the cell than there is on the inside of the cell, because 15% is much lower than that of 60%. So here's example problem 2. In example problem 2, there's cell A and cell B. Cell A has 20% glucose, cell B has 30% glucose. And there is a protein carrier facilitating and pumping the solute across. So what is the solute? The solute is glucose, and so glucose, if we put in our information, the movement of glucose with energy is then an active transport, because that is the solute. And so the data that we would want to analyze as we look at this is 20% solute on cell A, 30% solute in cell B, right? And so the concentration of solute is going to be irrespective of the gradient if it is active. And so in the same problem, if we consider which way or which solution has higher tonicity, cell B has 30% glucose, cell A has 20% glucose, and so in this regard, we can see that cell B, right, actually has higher tonicity, so cell B would be considered hypertonic to cell A. So endocytosis is the process of bringing substances into a cell, and we end up putting those substances in vesicles or vacuoles. And so phagocytosis is actually ingesting or consuming other substances or cells. And so there are pseudopods that form during the phagocytic pathway that will surround the external substance and allow us to bring that in and have it digested. In contrast, however, there is penocytosis, and penocytosis is the movement and bringing in of a liquid enclosed in a distinct microvilli. So here's an overall summary of transport mechanisms. The summary of transport mechanisms discusses the various types of diffusion through osmosis, or facilitated diffusion, or the movement of solutes, carrier-mediated active transport, and various translocation mechanisms. If a cell is in a concentrated glucose solution, and glucose is moving into the cell through a carrier protein, which is the correct answer? Facilitated diffusion would be the correct answer because it's moving through a carrier protein. It does not tell us if any energy is being expended, so that we cannot suggest that it would be active transport. So several environmental factors influence the survivability of a microbe. For example, a niche is the totality of the adaptations an organism must make to their habitat. So if an organism lives within a distinct niche, it is what specifications must they have to be able to survive that environment. And what we find, those niches are distinctively characterized based on temperature, oxygen requirements, pH, osmotic pressure, and barometric pressure. And so similar to us, microbes actually have a distinct temperature in which they prefer to thrive. The minimum temperature is the lowest temperature at which a microbe can still undergo metabolic processes and continue to live and double. The maximum temperature, however, is the highest temperature, so how hot a microbe can withstand and still undergo metabolic processes. And then finally, the optimum temperature is that perfect rate of growth at which a microbe can continue to thrive and will actually be most metabolically active. And so the adaptations that we have for various microbes, there are certain microbes that have been carefully adapted to live in very cold temperatures. These are called psychrophiles. So psychrophiles are pretty happy anywhere between 15 degrees and negative 15 degrees. And so they're capable of growth at 0. In this case, the optimum temperature for these are 15 degrees Celsius, but they can still grow as low with a minimum temperature of negative 15. Mesophiles, however, so most human pathogens fall under the category of mesophiles. They do so because our bodily temperature is 37 degrees Celsius. At 37 degrees Celsius, these mesophiles are pretty stinking happy, able to continue to thrive, cause infection and disease. And so they can, however, still thrive in much lower temperatures, so outside of the body. If they were left on individual surfaces, we can pick up microbes from doorknobs and things like that and continue to spread those types of infections. Thermophiles, so thermo meaning hot, so these guys have a greater temperature than 45 degrees Celsius, and so they can thrive pretty efficiently at very high temperatures. And so when we think about the other requirements, such as gas requirements, so gas is distinctly described as oxygen or carbon dioxide. And in this case, we're going to discuss oxygen. And so oxygen can be very toxic if you don't have the proper enzymes to detoxify it. So gas must be detoxified, or at least oxygen gas must be detoxified by superoxide dismutase or catalase. So any aerobic microbe must possess superoxide dismutase or catalase. And the reason is oxygen, as it's utilized, actually breaks down into things like peroxide or hydrogen peroxide or various radicals or superoxide ions, which are responsible for things like premature aging. If a microbe cannot detoxify the toxic oxygen components, it's going to be forced to live in an oxygen-free environment, so an anaerobic environment. And so aerobes utilize oxygen and detoxify oxygen. Obligate aerobes must live in an aerobic environment. They cannot grow without oxygen. A facultative anaerobe is a microbe that typically utilizes oxygen but can grow in its absence. And so a facultative anaerobe can grow in aerobic conditions as well as anaerobic conditions. Microaerophilic microbes, these guys require only a small amount of oxygen. So this is not regular atmospheric oxygen levels, but lower than normal, but they do not thrive in anaerobic conditions. An anaerobe is a microbe that does not use any oxygen. And then, of course, there's this obligate anaerobe, is a microbe that cannot in any way, shape, or form detoxify oxygen. And so if it were put in an oxygen-rich environment, it would die. And then there's distinct aerotolerant anaerobes. So these are microbes that actually prefer to live in anaerobic conditions. They don't utilize oxygen, but they possess the enzymes that are necessary to detoxify oxygen and continue to thrive in the presence of its toxic compounds. So here's an example of a distinct condition where various microbes have been grown. And so you can see here to the left, most of the bacteria has moved to the top of the tube. So what type of oxygen requirement would you suggest that this microbe has? Hopefully you decided that this microbe is an aerobe, or possibly even an obligate aerobe. Contrasting though, if you look here, you can see that there's this microbe that can grow distinctly all throughout the tube. So if the microbe can grow in the presence of oxygen and in the absence of oxygen down here deep within the tube, what type of microbe might it be? Facultative anaerobe would be the best choice because the microbe utilizes oxygen here but can also grow without it. And then here, this fourth tube, you can see that bacteria is growing well underneath the media all throughout the tube, but it's not growing at the top. The media is still very clear and not turbid. How would you describe this tube? So you could describe this tube as being an anaerobe, right? We're not fully sure if it's a obligate anaerobe in the sense that we don't know if the bacteria dies if it's here, but we can definitely tell that this bacteria does not want to grow in the presence of oxygen. So carbon dioxide, there are some microbes that require much higher carbon dioxide tensions than are present in the normal atmosphere. And so these microbes are considered a capnophile. So capnophiles are typically grown in anaerobe chambers where the carbon dioxide level is accelerated. So it can be grown in an anaerobe chamber. Sometimes we call these things a candle jar, where you actually light a candle, which sucks the additional oxygen out of the room as the flame burns, and then it actually leaves the excess carbon dioxide gas to higher quantities and higher surface tensions. The effects of pH. So along our body, we have all sorts of different levels of pH. So our skin is slightly acidic. We have a lot of acidity in our stomachs. The vaginal cavity is actually at a pH of about 2. And so we have microbes all over our body. We have bacteria covering nearly every surface. And so these microbes need to be able to thrive within extreme pH exchanges. So for example, if you have a microbe that can thrive in the gut, it has to be able to thrive the hydrochloric acid that is present within our stomachs, which arrives at a pH of 1. So any type of microbe that can survive a high acidic environment is considered an acidophile. Alternatively, an alkalinophile is a microbe that's going to be able to thrive at the entire opposite end of that pH scale. And so these guys are going to thrive in high pH, so 11 to 14, along the base side. And then the majority of microorganisms are going to land between 6 and 8. So these guys are neutrophiles. And so they prefer a moderate pH temperature. So they're probably not going to continually thrive in the vagina, but they can definitely thrive on most of the other bodily surfaces that we have. So osmotic pressure, or the osmotic gradient, is actually how we maintain the homeostasis of fluids throughout our body. So our kidneys actually process the movement of solute, or the reabsorption of solute or water, due to the osmotic gradient. And so there are some bacteria that can grow within a high concentration of salt. Those are considered osmotolerant. So osmotolerant molecules can tolerate the high concentration of salt, but they do not require it. In contrast to that, there are halophiles that indeed do require a high concentration of salt. And so if a halophile were placed into a non-saline rich environment, then the halophile would actually die because it needs that high concentration of salt. Is that of a barophile. So a barophile is a microbe that's going to live in the deep dwellings of the ocean, and it does so because of the high atmospheric pressure. Reflect on this briefly. If you've ever dove to the bottom of a pool and you feel the pressure along your head, or you feel your ears and the additional pressure, that's because of the barometric pressure underneath water. And so deep ocean prokaryotes can withstand this much higher pressure, but what happens is as those deep prokaryotes move to the surface, their cell walls are not adapted to atmospheric pressure, and then in turn they rupture. Clindomonas grows in Alaskan glaciers. It's a photosynthetic bacteria that causes these kind of red snow crusts. If this can live in Alaskan glaciers, what type of organism would it best be described as? A psychrophile, alkalinophile, microaerophile, osmotolerant, barophile. Does it have high pressure? No. Is there an osmotic gradient? No. Is there very small amounts of oxygen? We're not really sure based on the description of the glacier. Alkalinophile, there's no discussion of pH being high or basic, but we know that glaciers are cold, so best choice would be that of a psychrophile. The population or growth curve of a bacteria takes place due to a doubling time. So bacteria tend to double over a period of time. So some bacteria will double every 10 minutes, some will double every 15 minutes, and because of that doubling time, bacteria tend to double by a factor of 2 or result in an exponential growth. And so bacterial growth is often plotted on a log scale. And so here's an example problem. If E. coli has a doubling time of 20 minutes, if there's 5 cells at the beginning of the experiment, how many will be there after 3 hours? And so to begin this problem, my first advice for you is to start with time of 0. Always start with time of 0 so that you have the proper sequence by the time you are concluded. So E. coli, if you start with a time of 0 with 5 cells, 5 cells will become 10 cells after 20 minutes, 20 cells after 40 minutes, 40 cells after 60 minutes, 80 cells after 80 minutes, 160 cells after another doubling of 20 minutes, 320, 640, 1280, and then 2560. So the solution for this, 3 hours, which is actually 3 sets of 60 minutes, so 180 minutes, will be 2,560 cells of bacteria. And so you see that process here. So this is an example showing a doubling time of 60 minutes. And you can see that when you begin the experiment, there's no bacteria, but then eventually it's 2, 4, 7. That's not a perfect doubling. And then you go essentially forward and the doubling time and the pattern continue. And so the population growth curve starts out with an initial lag phase. So the lag phase is that early started growth period where the microbe has to get acclimated to the environment. My example for this is if you were to move to a new house. When you first move into your new house, all of your items are in boxes. And so first of all, before you're actually living efficiently or spreading out, you're going to have to unpack those boxes, and more often than not, that takes us a little bit of time. So the lag period is that lagging delay or that early pause that microbes have to get acclimated to their environment before they start reproducing. The exponential growth period, or the log period, because it is plotted on a logarithmic scale, is when the microbes are highly efficient. They are quickly doubling, they're replicating because there's lots of nutrients, it's plenty of space. So in the example of someone's actual house, you and your partner have moved into a 3-bedroom house. So now you're buying furniture for an office and furniture for a guest bedroom. But, as you continue to reproduce, possibly you have a child. And then later, maybe a few years down the road, you have several children. And at that point, your 3-bedroom house now is completely occupied. You don't have an office. You don't have a guest bedroom. You have children's rooms. And so at that point, you're now in a stationary phase. That stationary phase is because the rate of cell growth equals the rate of cell death. So the space within your home is completely occupied, so you're probably not considering having a third child because the third child does not have a bedroom in your current theoretical 3-bedroom home. And so in the stationary phase, essentially the rate of death and the rate of nutrient depletion is kind of at this steady pace, and it cycles through. But eventually there is a buildup of factors that cause the cells to die. So there's only so much space, there's only so much nutrients. And, in this scenario, we're not going to kill you off in your little imaginary 3-bedroom house with 2.5 children. Instead, you would probably leave that house and go buy a new one. So for the death phase with microbes, they don't have the option to just pick up and move, well, unless they're free-living microbes. And in that case, they also would move to a new location. If they stay within that concentrated environment, however, the absence of nutrients would cause them to die. Now, I want you to reflect back to chapter 4 when we were talking about endospores. What would happen if you have a microbe that is undergoing the death phase, but is a microbe that produces endospores? Will this bacteria die? The answer is no. The bacteria will not die, the vegetative cell will die, and the endospores will remain and will allow for a germination stage if nutrients ever become available in the future.